Advantages of genetic engineering, unveiling the benefits of genetic manipulation. 1. Improved crop yield and quality. Genetic engineering offers the potential to enhance crop yield and improve their nutritional content. By introducing specific genes into plants, scientists can confer traits such as resistance to pests, diseases, and environmental stresses. This technology allows for the development of crops that produce higher yields, withstand harsh conditions, and have increased nutritional value, thereby addressing food security concerns and improving the overall quality of agricultural produce. 2. Disease resistance in plants and animals. Genetic engineering enables the introduction of genes that provide resistance against harmful pathogens in plants and animals. This can help mitigate the impact of devastating diseases that can destroy entire crops or cause significant losses in livestock. By genetically modifying organisms to possess innate resistance, farmers and growers can reduce the reliance on chemical pesticides and antibiotics, promoting sustainable agricultural practices and reducing the risk of harmful residues in the food chain. 3. Medical advancements and therapeutic applications. Genetic engineering has revolutionized medical research and therapeutic applications. Scientists can modify the genetic material of organisms, including humans, to study the function of specific genes and their role in diseases. This understanding has led to the development of targeted therapies, such as gene therapy, where defective genes are replaced or corrected, offering potential cures for genetic disorders. Additionally, genetic engineering has facilitated the production of pharmaceutical proteins, vaccines, and other medical treatments, improving healthcare outcomes. 4. Environmental sustainability. Genetic engineering contributes to environmental sustainability by offering innovative solutions to pressing environmental challenges. For example, genetically modified organisms, GMOs, can be engineered to produce enzymes that aid in the breakdown of pollutants, facilitating bioremediation. Additionally, genetically modified crops can be designed to require fewer chemical inputs, such as pesticides and fertilizers, reducing their environmental impact. This technology also holds promise for developing renewable energy sources by enhancing the efficiency of biofuel production. 5. Industrial applications and biotechnology. Genetic engineering plays a crucial role in industrial applications and biotechnology. Through the manipulation of microorganisms, scientists can create more efficient and cost-effective processes for the production of biofuels, pharmaceuticals, enzymes, and other valuable chemicals. This not only improves the efficiency of manufacturing processes but also reduces the dependence on non-renewable resources and minimizes waste production, making industries more sustainable and environmentally friendly. 6. Conservation and preservation of endangered species. Genetic engineering offers possibilities for conserving and preserving endangered species. By using assisted reproductive technologies and genetic manipulation, scientists can work towards increasing the genetic diversity and reproductive success of endangered populations. This can help prevent the extinction of threatened species and restore ecological balance in delicate ecosystems. 7. Scientific research and understanding of genetics. Genetic engineering has significantly advanced scientific research and our understanding of genetics. By manipulating genes in model organisms, scientists can uncover valuable insights into gene function, development, and evolutionary processes. This knowledge expands our understanding of life and provides a foundation for further discoveries and innovations in various scientific fields. In conclusion, genetic engineering presents numerous advantages that span across agriculture, medicine, industry, and conservation. These advancements have the potential to address global challenges, improve human well-being, and pave the way for a more sustainable future. Advancements in agriculture, enhanced crop yields and resilience to pests and diseases. Genetic engineering has emerged as a powerful tool in agriculture, offering substantial benefits such as improved crop yields and enhanced resistance to pests and diseases. By employing genetic manipulation techniques, scientists can introduce specific genes into plants, resulting in valuable traits that contribute to higher productivity and increased resilience. Crop yields play a vital role in ensuring global food security. With the world's population steadily increasing, it is crucial to develop sustainable agricultural practices that can meet the growing demand for food. Genetic engineering allows scientists to modify crops to achieve improved productivity. This can be achieved by enhancing the plant's ability to utilize nutrients efficiently, optimizing photosynthesis, or extending the growing season. By introducing genes responsible for these traits, crop plants can produce higher yields, providing a significant contribution to addressing food shortages. In addition to higher yields, genetic engineering enables the development of crops with enhanced resistance to pests and diseases. Traditional methods of pest control often rely on the use of chemical pesticides, which can have negative environmental impacts and pose risks to human health. By incorporating genes from naturally resistant organisms, scientists can confer pest resistance to crop plants, reducing the reliance on harmful chemicals. This not only minimizes the environmental impact but also promotes sustainable agricultural practices. Disease outbreaks can devastate crops, leading to significant economic losses and food shortages. Genetic engineering offers a solution by equipping plants with genes that confer resistance to specific pathogens. By introducing genes that produce proteins capable of combating diseases, crops can effectively defend themselves against harmful pathogens, reducing the need for excessive pesticide applications. This approach provides a more targeted and environmentally friendly method of disease control, benefiting both farmers and consumers. Furthermore, genetic engineering allows for the development of crops with increased tolerance to environmental stresses. Climate change presents challenges such as drought, extreme temperatures, and salinity, which can severely impact crop productivity. By introducing genes that enhance stress tolerance, plants can better withstand adverse conditions, ensuring more reliable yields even in challenging environments. This is particularly crucial in regions prone to climatic fluctuations, where the ability to cultivate crops with improved resilience can make a significant difference in food production. Overall, genetic engineering plays a crucial role in improving crop yields and combating agricultural challenges. By harnessing the power of genetic manipulation, scientists can develop crops that are more productive, resilient to pests and diseases, and capable of withstanding environmental stresses. These advancements have the potential to revolutionize agriculture, ensuring food security, reducing environmental impacts, and contributing to the sustainable development of our planet. Revolutionizing agriculture, genetic engineering for enhanced crop resistance and increased food production. Genetic engineering has opened up new possibilities in agriculture by enabling scientists to selectively introduce specific genes into plants. This process empowers crops with enhanced resistance to pests, diseases, and unfavorable environmental conditions, ultimately resulting in a multitude of benefits such as increased crop yields, minimized losses, and improved global food production. The ability to introduce specific genes into plants through genetic engineering has paved the way for developing crop varieties with heightened resistance to pests and diseases. By incorporating genes that encode natural defense mechanisms found in other organisms, scientists can bolster the plant's ability to fend off attacks from insects, viruses, bacteria, and fungi. This fortification reduces crop losses caused by devastating pests and diseases, preserving the overall quality and quantity of agricultural output. As a result, genetic engineering contributes significantly to safeguarding global food security by ensuring a more abundant and stable supply of food. Additionally, genetic engineering equips crops with traits that enhance their ability to withstand adverse environmental conditions. Climate change has brought about more frequent and severe challenges, such as drought, extreme temperatures, and soil salinity, which can severely impact crop growth and yield. By introducing genes that confer tolerance to these stresses, scientists can help crops thrive in otherwise inhospitable environments. This newfound resilience enables plants to maintain their growth and productivity even under suboptimal conditions, ultimately increasing food production in regions that would otherwise struggle to support agricultural activities. The introduction of specific genes into plants through genetic engineering also offers the potential for improving the nutritional content of crops. scientists can introduce genes that enhance the synthesis of essential nutrients, such as vitamins, minerals, and amino acids. This biofortification strategy aims to address nutritional deficiencies prevalent in certain regions, where access to diverse and nutrient-rich diets may be limited. By developing crops with enhanced nutritional profiles, genetic engineering contributes to improving the overall health and well-being of populations worldwide. Moreover, genetic engineering provides a precise and targeted approach to crop improvement compared to traditional breeding methods. It allows scientists to select and introduce specific genes with known functions and desired traits, accelerating the process of crop improvement. This efficiency results in a faster turnaround time for developing improved crop varieties, reducing the time required to bring valuable traits to farmers and consumers. In summary, genetic engineering has revolutionized agriculture by enabling the introduction of specific genes into plants, enhancing their resistance to pests, diseases, and adverse environmental conditions. This breakthrough technology contributes to increased crop yields, reduced crop losses, and improved food production, playing a vital role in addressing global food security challenges. By harnessing the power of genetic manipulation, scientists continue to push the boundaries of crop improvement, creating a more sustainable and resilient agricultural system for the benefit of humanity. Biofortification, elevating the nutritional value of food for improved health. Biofortification is a powerful technique that leverages genetic engineering to enhance the nutritional value of food, offering a range of health benefits to individuals and populations. By selectively introducing specific genes into crops, scientists can boost the content of essential nutrients, such as vitamins, minerals, and antioxidants, in the edible parts of plants. The process of biofortification begins with identifying genes responsible for the synthesis or accumulation of desired nutrients. For example, scientists may introduce genes that enhance the production of vitamin A in rice, iron and zinc in wheat, or folate in maize. These genes can be sourced from other organisms or identified within the same crop species. Once the genes are incorporated into the crop plants, they enable the production of higher levels of targeted nutrients. Biofortified crops hold tremendous potential in addressing widespread nutrient deficiencies and associated health problems. Many regions of the world suffer from inadequate intake of key nutrients, leading to malnutrition and various deficiencies. By cultivating and consuming biofortified crops, populations can significantly improve their nutrient intake, promoting better overall health and reducing the prevalence of deficiencies. For instance, vitamin A deficiency is a major global public health concern, particularly in developing countries. Insufficient intake of this essential nutrient can lead to impaired vision, weakened immune function, and increased susceptibility to infectious diseases. By biofortifying staple crops such as rice, which are widely consumed by vulnerable populations, with increased levels of vitamin A precursors such as beta-carotene, genetic engineering offers a sustainable solution to combating this deficiency. Biofortification also contributes to combating iron and zinc deficiencies, which are prevalent worldwide and particularly affect women and children. Insufficient intake of these minerals can lead to anemia, impaired cognitive development, and compromised immune function. Through biofortification, staple crops like wheat, maize, and beans can be fortified with higher levels of iron and zinc, helping to address these widespread deficiencies and improve the health and well-being of communities. Furthermore, biofortification offers a promising approach to address nutrient deficiencies in populations where access to a diverse and balanced diet is limited. By improving the nutritional content of commonly consumed crops, genetic engineering provides an effective and sustainable solution that can reach individuals who may have limited access to other sources of essential nutrients. It is important to note that biofortification complements other strategies to combat malnutrition, such as dietary diversification and micronutrient supplementation. It offers a long-term and sustainable solution by integrating enhanced nutritional value directly into the food crops themselves, ensuring continuous availability and affordability for populations. In conclusion, biofortification through genetic engineering represents a powerful tool for elevating the nutritional value of food crops. By enhancing the content of essential nutrients, biofortified crops hold the potential to address widespread nutrient deficiencies, improve overall health, and contribute to the well-being of populations worldwide. Continued research and adoption of biofortification can play a vital role in combating malnutrition and promoting a healthier future for generations to come. Biofortification, enhancing nutritional content of food crops for public health improvement. Biofortification, a process facilitated by genetic engineering, offers a promising avenue to enhance the nutritional content of food crops. By selectively introducing genes that boost the levels of vital nutrients such as vitamins, minerals, and proteins, biofortification holds the potential to address nutrient deficiencies and significantly improve public health. This approach is particularly beneficial in regions where specific nutrients are lacking in diets, providing a sustainable solution to enhance the nutritional value of commonly consumed crops. The process of biofortification involves identifying and incorporating genes responsible for the synthesis or accumulation of targeted nutrients into crops. For example, scientists may introduce genes that enhance the production of vitamin A, iron, zinc, or protein in staple crops. These genes can be sourced from related species or even from different organisms with high nutrient content. By integrating these genes into crop plants, the enhanced nutrient levels can be effectively achieved. The utilization of biofortified crops offers a compelling strategy to combat nutrient deficiencies, which are prevalent in many regions of the world. Insufficient intake of essential nutrients can lead to various health issues and impairments. For example, vitamin A deficiency can cause visual impairments and weakened immune systems, particularly affecting vulnerable populations in developing countries. By biofortifying staple crops, such as rice or sweet potatoes, with genes that increase the production of vitamin A precursors, genetic engineering provides a sustainable approach to addressing this deficiency and promoting better health outcomes. Iron and zinc deficiencies are also major global health concerns, especially among women and children. Insufficient intake of these minerals can lead to anemia, impaired cognitive development, and compromised immune function. Through biofortification, crop plants like wheat, maize, and beans can be modified to accumulate higher levels of iron and zinc, effectively increasing the nutritional value of these staple foods and combating nutrient deficiencies. Biofortification offers a practical and cost-effective solution to improve public health, particularly in regions where access to diverse and balanced diets may be limited. By enhancing the nutritional content of commonly consumed crops, genetic engineering provides a sustainable means to address nutrient deficiencies and improve overall dietary quality. It offers an opportunity to bridge the gap between nutritional requirements and actual nutrient intake, particularly in populations that may have limited access to alternative sources of essential nutrients. Moreover, biofortification complements other strategies aimed at improving nutrition, such as dietary diversification and micronutrient supplementation. By directly incorporating enhanced nutrient content into the crops themselves, biofortification ensures a continuous supply of these vital nutrients without requiring additional interventions or resources. It is a long-term approach that can positively impact public health by making nutrient-rich foods more readily available and affordable to diverse populations. In conclusion, genetic engineering through biofortification enables the enhancement of nutritional content in food crops, addressing nutrient deficiencies and promoting public health improvement. By introducing genes that increase the levels of essential nutrients, biofortified crops have the potential to fill the nutrient gaps in diets, particularly in regions where specific nutrients are lacking. Continued research and adoption of biofortification can contribute to a healthier future, ensuring that communities have access to nutrient-rich food sources and reducing the burden of nutrient-related health issues on a global scale. Revolutionizing medicine, genetic engineering for pharmaceutical production and advanced medical treatments. The field of genetic engineering has ushered in a new era of medical advancements, enabling the production of pharmaceuticals and the development of cutting-edge medical treatments. By harnessing the power of genetic manipulation, scientists can engineer organisms to produce valuable pharmaceutical proteins and create innovative therapies that offer hope for treating various diseases and improving healthcare outcomes. 1. Production of pharmaceuticals. Genetic engineering plays a pivotal role in the production of pharmaceuticals. Traditionally, these drugs were obtained through chemical synthesis or extraction from natural sources, which often proved challenging, costly, and time-consuming. However, genetic engineering techniques have revolutionized this process by enabling the production of pharmaceutical proteins using recombinant DNA technology. Through genetic engineering, scientists can introduce genes encoding specific therapeutic proteins into microorganisms, such as bacteria or yeast, or even into plants and animals. These genetically modified organisms act as bioreactors, efficiently synthesizing large quantities of the desired proteins. This approach offers several advantages, including high yields, cost-effectiveness, and the ability to customize and optimize protein production. The production of pharmaceuticals through genetic engineering has transformed the treatment landscape for various diseases. Insulin, for example, which was traditionally extracted from animal sources, can now be produced using recombinant DNA technology. This has ensured a stable and sustainable supply of this life-saving hormone for individuals with diabetes. 2. Advanced medical treatments. Genetic engineering has revolutionized medical treatments, opening up new frontiers for innovative therapies. One notable application is gene therapy, which aims to treat or cure genetic disorders by introducing healthy genes into a patient's cells. By utilizing viral vectors or other delivery mechanisms, genetic engineers can target specific cells and tissues, correcting genetic abnormalities at their source. Gene therapy holds immense promise for treating a wide range of diseases, including inherited disorders, certain types of cancers, and even some viral infections. For example, in cases of severe combined immunodeficiency, SCID, also known as bubble boy disease, gene therapy has shown remarkable success in restoring immune function by introducing functional copies of defective genes. Additionally, genetic engineering plays a crucial role in the development of novel medical treatments, such as personalized medicine and immunotherapies. By understanding the genetic basis of diseases, scientists can design targeted therapies that specifically address the underlying molecular mechanisms. This approach offers the potential for more precise and effective treatments, minimizing side effects and improving patient outcomes. Furthermore, genetic engineering enables the production of therapeutic antibodies, which have revolutionized the treatment of various diseases, including cancer, autoimmune disorders, and infectious diseases. Through genetic manipulation, scientists can engineer cells to produce specific antibodies that target disease-causing agents, enhancing the body's immune response and aiding in disease eradication. In conclusion, genetic engineering has transformed the production of pharmaceuticals and revolutionized medical treatments. By employing genetic manipulation techniques, scientists can engineer organisms to produce pharmaceutical proteins efficiently. Moreover, genetic engineering enables the development of advanced medical treatments, such as gene therapy, personalized medicine, and immunotherapies, offering promising avenues for treating genetic disorders, cancers, and various diseases. These advancements hold tremendous potential for improving healthcare outcomes, enhancing the quality of life for patients, and shaping the future of medicine. Revolutionizing pharmaceutical production and disease treatment, genetic engineering's role. Genetic engineering has become a game-changer in the production of valuable pharmaceuticals and the development of advanced medical treatments. By utilizing genetically modified organisms, scientists can tap into their potential to produce essential medications and contribute to disease prevention and treatment in remarkable ways. 1. Production of pharmaceuticals. Genetic engineering plays a pivotal role in the production of pharmaceuticals, offering efficient and sustainable methods for generating vital drugs. By harnessing the capabilities of genetically modified organisms, scientists can engineer them to produce therapeutic proteins and other compounds crucial for medical treatments. One notable example is the production of insulin. Previously, insulin extraction from animal sources was the primary method. However, genetic engineering has revolutionized this process. Scientists can now introduce the genes responsible for insulin production into bacteria, such as Escherichia coli. These genetically modified bacteria act as bioreactors, synthesizing insulin in large quantities. This method ensures a reliable and abundant supply of this life-sustaining hormone for individuals managing diabetes. Furthermore, genetic engineering has facilitated the development of other pharmaceuticals beyond insulin. By introducing genes that encode specific therapeutic proteins into microorganisms, scientists can utilize them as efficient protein factories. This approach offers numerous advantages, including scalability, cost-effectiveness, and the ability to customize protein production to meet specific needs. 2. Development of vaccines. Genetic engineering also plays a critical role in the development of vaccines, revolutionizing disease prevention and treatment. Vaccines are essential for safeguarding public health by stimulating the immune system to recognize and fight specific pathogens. Through genetic engineering, scientists can create vaccines that are safer, more effective, and more easily produced. One method involves using genetically modified organisms, such as bacteria or yeast, to produce key components of vaccines. For example, the hepatitis B vaccine is produced by introducing the viral gene into yeast, which then produces the viral surface antigen used in the vaccine. This process provides a reliable and cost-effective means of large-scale vaccine production. Furthermore, genetic engineering has enabled the development of novel vaccines, including DNA vaccines and viral vector-based vaccines. DNA vaccines involve introducing specific genes encoding pathogen antigens directly into the body's cells, triggering an immune response. Viral vector-based vaccines use genetically engineered viruses to deliver antigens and stimulate an immune response. These advancements in vaccine development have opened up new possibilities for preventing and treating a wide range of diseases, including infectious diseases and certain types of cancer. In conclusion, genetic engineering has revolutionized the production of pharmaceuticals and the development of vaccines. By utilizing genetically modified organisms, scientists can efficiently produce vital medications, such as insulin, and contribute to improved disease prevention and treatment. The versatility and precision offered by genetic engineering hold immense potential for the pharmaceutical industry and the field of medicine as a whole, shaping a future where innovative therapies and effective vaccines are more readily available to enhance human health and well-being. Harnessing the potential of genetically modified organisms, GMOs, for environmental remediation. The development and utilization of genetically modified organisms, GMOs, have emerged as a promising approach for environmental remediation, offering innovative solutions to address various environmental challenges. By genetically engineering organisms, scientists can enhance their ability to break down pollutants, mitigate contamination, and restore ecological balance in a more efficient and sustainable manner. 1. Environmental pollutant degradation. Genetic engineering enables the modification of organisms to produce enzymes or metabolic pathways that aid in the breakdown and detoxification of environmental pollutants. This approach has significant potential for remediating contaminated soil, water, and air. For example, certain bacteria can be genetically modified to produce enzymes capable of breaking down toxic substances such as polychlorinated biphenyls, PCBs, petroleum hydrocarbons, or pesticides. These modified bacteria can be introduced into polluted environments to facilitate the degradation of these pollutants, reducing their environmental impact and promoting the restoration of ecosystems. 2. Bioremediation and biodegradation. GMOs can also be engineered to possess enhanced bioremediation and biodegradation capabilities. By introducing genes that enable organisms to better metabolize or assimilate specific contaminants, scientists can create GMOs with improved abilities to remove pollutants from the environment. One example is the engineering of plants to express enzymes that break down pollutants present in the soil. These modified plants, often referred to as phytoremediation plants, can absorb contaminants through their roots and metabolize or store them, effectively reducing soil contamination. This approach has shown promise in remediating heavy metal contamination and organic pollutants, mitigating their adverse effects on ecosystems and human health. 3. Waste treatment and resource recovery. GMOs can also play a role in waste treatment and resource recovery processes. By genetically engineering microorganisms, scientists can optimize their metabolic pathways to efficiently break down and convert waste materials into valuable resources. For instance, genetically modified bacteria can be utilized to break down organic waste in wastewater treatment plants. These bacteria can metabolize organic pollutants, converting them into simpler and less harmful compounds. This not only helps in wastewater treatment but also offers the potential for resource recovery, as the byproducts of microbial metabolism can be harnessed as energy sources or nutrient-rich fertilizers. 4. Improved agricultural practices. Genetic engineering has the potential to enhance agricultural practices and reduce environmental impacts. GMOs can be engineered to possess traits that promote sustainable farming, such as increased nutrient use efficiency, resistance to pests and diseases, and tolerance to environmental stresses. For example, genetically modified crops can be designed to require fewer chemical inputs, such as pesticides and fertilizers. By incorporating genes that confer resistance to pests or enhance nutrient uptake, farmers can reduce the use of harmful chemicals, minimizing their impact on the environment and improving soil and water quality. In summary, genetic engineering offers promising opportunities for environmental remediation. By developing genetically modified organisms, GMOs, with enhanced capabilities, scientists can contribute to the degradation of pollutants, the restoration of ecosystems, and the implementation of more sustainable agricultural practices. However, it is essential to consider potential risks and conduct thorough assessments to ensure the responsible and safe deployment of GMOs for environmental purposes. Harnessing the potential of genetic engineering for environmental cleanup. Genetic engineering has opened up possibilities for utilizing genetically modified organisms, GMOs, in environmental cleanup efforts, presenting innovative solutions to address various environmental challenges. By leveraging genetic manipulation techniques, scientists can modify organisms to possess traits that enable them to degrade harmful pollutants, absorb and accumulate heavy metals, and contribute to the restoration of polluted ecosystems. This approach holds promise in addressing environmental challenges and promoting sustainable practices. 1. Genetic modification for pollutant degradation. Genetic engineering allows for the modification of organisms to produce enzymes or metabolic pathways that aid in the breakdown and detoxification of harmful pollutants. Certain bacteria, for example, can be genetically engineered to produce specific enzymes capable of degrading contaminants like polychlorinated biphenyls, PCBs, petroleum hydrocarbons, or pesticides. These modified bacteria can then be introduced into polluted environments to facilitate the degradation of these pollutants, minimizing their impact on ecosystems and human health. 2. GMOs for heavy metal absorption and accumulation. GMOs can also be engineered to possess enhanced capabilities for absorbing and accumulating heavy metals, aiding in the remediation of metal-contaminated sites. Certain plants, referred to as hyperaccumulators, can be genetically modified to increase their ability to absorb heavy metals from the soil through their roots. These GMO plants can then be utilized to remove and accumulate heavy metals, thereby reducing soil contamination and mitigating the adverse effects of metal pollution on ecosystems. 3. Restoration of polluted ecosystems. By using genetic engineering, organisms can be modified to possess traits that contribute to the restoration of polluted ecosystems. GMOs can aid in the cleanup of contaminated sites, helping to restore ecological balance and promote the recovery of native flora and fauna. 4. Potential for sustainable environmental practices. Genetic engineering holds the potential to promote sustainable environmental practices by reducing the need for harmful chemical interventions and enhancing resource efficiency. By engineering crops with increased resistance to pests and diseases, farmers can reduce their reliance on chemical pesticides, minimizing their environmental impact. Additionally, genetic modification can be employed to enhance nutrient uptake efficiency in crops, reducing the need for excessive fertilizers and preventing nutrient runoff that can pollute water bodies. In summary, genetic engineering offers promising avenues for utilizing GMOs in environmental cleanup initiatives. By modifying organisms, scientists can develop GMOs capable of degrading pollutants, absorbing heavy metals, and aiding in the restoration of polluted ecosystems. This approach not only addresses environmental challenges but also has the potential to promote sustainable practices by reducing chemical inputs and improving resource efficiency. However, careful risk assessment, regulatory oversight, and responsible deployment are crucial to ensuring the safe and effective use of GMOs for environmental purposes. Exploring the drawbacks of genetic engineering. 1. Ethical and moral concerns. Genetic engineering raises ethical and moral concerns regarding the manipulation of living organisms. Critics argue that altering the genetic makeup of organisms, especially in the context of humans or animals, may infringe upon the principles of natural selection and autonomy. There are debates surrounding the potential risks and unintended consequences of modifying organisms in ways that may have far-reaching and unpredictable effects on ecosystems, biodiversity, and the intrinsic value of life. 2. Environmental risks. Genetic engineering poses potential risks to the environment. Genetically modified organisms, GMOs, have the potential to interact with native species, potentially leading to the disruption of ecosystems and biodiversity. Concerns arise regarding the possibility of unintended gene flow between GMOs and wild relatives, which may result in the spread of modified traits to unintended populations. Furthermore, the long-term ecological impacts of genetically modified crops and organisms are still not fully understood, making it challenging to accurately assess their potential risks and consequences. 3. Health and safety concerns. Another critical consideration is the potential health and safety risks associated with genetic engineering. While stringent regulations are in place to ensure the safety of genetically modified organisms for human consumption, concerns remain about potential allergenicity and toxicity. The introduction of novel genes or the alteration of existing ones might inadvertently introduce allergenic substances or other unintended effects that could pose risks to human health. Vigilant testing and rigorous safety assessments are essential to address these concerns and ensure the protection of public health. 4. Socioeconomic implications. Genetic engineering can have significant socioeconomic implications, particularly in agriculture. Critics argue that the widespread adoption of genetically modified crops could lead to increased corporate control over the agricultural sector. Patents and intellectual property rights associated with genetically modified seeds and organisms may limit farmers' autonomy and access to traditional seed varieties, potentially perpetuating dependency on a few major corporations. Additionally, concerns exist about the potential for increased social and economic inequality, as smaller farmers may struggle to compete with larger agribusinesses that have the resources to adopt and benefit from genetically modified technologies. 5. Unintended consequences and unpredictability. The complex nature of genetics and biological systems introduces the potential for unintended consequences and unpredictability in genetic engineering. Despite extensive testing and regulation, it is challenging to foresee all possible outcomes and interactions resulting from genetic modifications. Unintended effects can emerge due to interactions between modified genes and other genetic elements, potentially leading to unexpected outcomes or unforeseen environmental or health risks. 6. Regulatory and oversight challenges. Genetic engineering requires robust regulatory frameworks and oversight to ensure responsible and safe practices. However, developing and implementing effective regulations can be challenging. Balancing innovation and technological advancements with the need for precaution and risk assessment is a complex task. Inadequate regulation or oversight may result in inadequate safety precautions, inadequate labeling, or insufficient monitoring of the long-term effects of genetically modified organisms. In conclusion, genetic engineering brings with it a set of disadvantages and concerns that need to be carefully addressed. Ethical considerations, environmental risks, health and safety concerns, socioeconomic implications, unpredictability, and regulatory challenges all require thoughtful evaluation and appropriate safeguards to ensure the responsible and safe application of genetic engineering technologies. Striking a balance between the potential benefits and the associated drawbacks is crucial for informed decision-making and sustainable development. Ethical considerations, manipulation of living organisms and unintended consequences. The field of genetic engineering raises significant ethical concerns surrounding the manipulation of living organisms, particularly when it comes to humans, animals, and the potential for unintended consequences. These ethical considerations delve into the core principles of natural selection, autonomy, and the potential risks and consequences associated with modifying the genetic makeup of organisms. 1. Manipulation of living organisms. One of the key ethical concerns is the fundamental question of whether it is ethically justifiable to alter the genetic material of living organisms. Critics argue that genetic engineering may disrupt the principles of natural selection, as the process involves intentional modification of genes rather than relying on natural evolutionary processes. This raises questions about the balance between human intervention and the integrity of natural systems. Moreover, genetic engineering in the context of humans presents complex ethical considerations. The ability to modify human genes raises concerns about the potential for creating designer babies or altering hereditary traits, which may have far-reaching societal and ethical implications. Discussions around the ethical boundaries of genetic modification in humans often center on issues of consent, equity, and the potential for exacerbating existing social inequalities. 2. Unintended consequences. The unpredictability of genetic engineering and the potential for unintended consequences pose ethical concerns. Genetic modifications may lead to unintended effects, which could impact the target organism or have unforeseen consequences on ecosystems and biodiversity. Despite rigorous testing and safety protocols, the complexity of biological systems makes it difficult to anticipate all potential interactions and outcomes resulting from genetic modifications. Unintended consequences can arise from various factors, including gene interactions, gene flow between modified organisms and wild relatives, or ecological disruptions caused by genetically modified organisms introduced into ecosystems. These uncertainties highlight the need for careful risk assessment, ongoing monitoring, and transparent communication to address ethical concerns and mitigate potential negative impacts. Ethical considerations in genetic engineering call for a balance between scientific progress and responsible innovation. Deliberate discussions involving stakeholders from various fields, including ethics, philosophy, and public engagement, are necessary to ensure that ethical frameworks and guidelines are in place to guide the ethical and responsible application of genetic engineering technologies. Ethical considerations, deliberate modification of living organisms and potential risks. The field of genetic engineering triggers profound ethical questions regarding the intentional modification of living organisms and the potential risks that come with it. These ethical considerations delve into the moral implications of altering the genetic makeup of organisms, especially when it involves complex traits or genetic modifications in humans. 1. Modification of living organisms. Genetic engineering raises ethical concerns about the deliberate alteration of the genetic material of living organisms. Critics argue that such interventions may disrupt the principles of natural evolution and the inherent value of organisms. These concerns touch upon philosophical debates surrounding the boundaries of human intervention in the natural world and the potential consequences of deviating from natural selection processes. The deliberate modification of organisms also raises ethical questions about the potential consequences for ecosystems and biodiversity. Introducing genetically modified organisms into the environment could lead to unintended ecological disruptions and harm to native species. Striking a balance between the benefits and potential risks is essential to ensure responsible and ethical practices. 2. Ethical implications in human genetic modification. Genetic engineering technologies have the potential to modify the genetic traits of humans, introducing complex ethical considerations. The ability to alter the genetic makeup of individuals raises questions about the moral boundaries of human intervention and the potential consequences of manipulating human heredity. One prominent concern is the development of designer babies, where genetic modifications are made to enhance specific traits or characteristics. This raises ethical dilemmas surrounding issues of equity, social justice, and the potential for exacerbating existing inequalities. It also prompts discussions about the limits of parental autonomy and the potential impact on future generations. Furthermore, genetic modifications in humans can carry potential health and safety risks. Despite extensive testing and regulations, there remains the possibility of unintended consequences and unforeseen health implications associated with genetic alterations. Ethical frameworks must address these concerns and ensure that thorough risk assessments and transparent communication are in place to protect the well-being and autonomy of individuals. Addressing ethical concerns. Addressing the ethical concerns associated with genetic engineering requires comprehensive discussions involving experts from diverse fields, including ethics, philosophy, science, and public engagement. These discussions aim to establish ethical guidelines, frameworks, and regulations to guide responsible and ethical practices. Ethical considerations emphasize the importance of informed consent, transparency, and the equitable distribution of benefits and risks associated with genetic engineering technologies. Public engagement and dialogue enable broader participation and ensure that societal values, concerns, and perspectives are incorporated into decision-making processes. Ultimately, a careful balance must be struck between scientific advancement, societal values, and responsible innovation to navigate the ethical complexities of genetic engineering. By acknowledging and addressing ethical concerns, we can strive for the responsible and ethical application of genetic engineering technologies, promoting the well-being of both humans and the natural world. Ecological risks, gene flow to wild populations and disruption of natural ecosystems. The application of genetic engineering carries ecological risks that stem from the potential for gene flow to wild populations and the disruption of natural ecosystems. These risks raise concerns about the unintended consequences of genetically modified organisms, GMOs, and their potential impact on biodiversity and ecological balance. 1. Gene flow to wild populations. Gene flow refers to the transfer of genetic material from genetically modified organisms to their wild relatives or other related species. This phenomenon can occur through various mechanisms such as pollination, seed dispersal, or hybridization. The introduction of genetically modified traits to wild populations raises concerns about potential ecological consequences. If genetically modified traits are transferred to wild populations, it may result in the spread of those traits beyond intended boundaries. This can have unforeseen impacts on the fitness, adaptation, and ecological interactions of wild organisms. Concerns arise regarding the potential loss of genetic diversity, the disruption of natural evolutionary processes, and the alteration of ecosystems in ways that are difficult to predict. 2. Disruption of natural ecosystems. The release of genetically modified organisms into natural ecosystems carries the risk of disrupting ecological dynamics. Ecosystems are complex networks of interdependent species, and any introduction of genetically modified organisms has the potential to impact the interactions and balance within these ecosystems. For instance, genetically modified crops may exhibit altered characteristics such as increased competitiveness or resistance to pests. If these modified crops escape cultivation and establish themselves in the wild, they could outcompete native species or disrupt ecological relationships. Such disruptions can have cascading effects on biodiversity, including impacts on native flora and fauna, as well as the ecological services that ecosystems provide. 3. Unintended ecological consequences. Despite rigorous testing and risk assessment, the complexity of ecological systems makes it challenging to predict all potential ecological consequences of genetic engineering. The introduction of genetically modified organisms into the environment may lead to unintended ecological interactions, unexpected spread of modified traits, or unforeseen ecological disruptions. For example, modifications aimed at conferring resistance to specific pests or diseases in crops might inadvertently impact non-target organisms, such as beneficial insects or soil microorganisms, with potential implications for ecological balance and ecosystem functioning. Addressing ecological risks. Addressing the ecological risks associated with genetic engineering requires a comprehensive and precautionary approach. Risk assessments, ecological studies, and monitoring programs are essential to evaluate the potential impacts of genetically modified organisms on wild populations and ecosystems. Strict containment measures and regulations can help prevent the unintended release of genetically modified organisms into natural environments. Furthermore, promoting biodiversity conservation, ecosystem resilience, and the restoration of natural habitats can contribute to mitigating ecological risks. Emphasizing ecosystem-based approaches, such as agroecology and ecological restoration, can provide sustainable alternatives that minimize the reliance on genetic modification and focus on promoting biodiversity and ecological health. By adopting a holistic approach that combines rigorous risk assessment, robust regulations, ecological monitoring, and the preservation of natural ecosystems, it is possible to navigate the ecological risks associated with genetic engineering and ensure the responsible and sustainable application of this technology. Ecological risks, gene flow and impacts on natural ecosystems and biodiversity. The introduction of genetically modified organisms, GMOs, into the environment poses ecological risks, particularly through the process of gene flow, where modified genes can spread to wild populations via crossbreeding. These risks encompass unpredictable consequences that may include the disruption of natural ecosystems and the loss of biodiversity. Gene flow and the spread of modified genes. Gene flow occurs when genes from genetically modified organisms are transferred to wild populations or related species. This transfer can happen through various mechanisms such as pollen dispersal, seed dissemination, or hybridization. When modified genes enter wild populations, it raises concerns about the potential spread of these genes beyond their intended boundaries. The unpredictable consequences. The introduction of modified genes into wild populations can have unpredictable consequences for ecosystems and biodiversity. The expression of modified traits in wild organisms can disrupt natural evolutionary processes, alter ecological interactions, and influence the adaptation and fitness of affected species. These unforeseen impacts may lead to shifts in species composition, changes in ecological dynamics, and a potential loss of genetic diversity. Disruption of natural ecosystems. The presence of genetically modified organisms in natural ecosystems carries the risk of disrupting their ecological balance. Ecosystems thrive on intricate relationships between species, and the introduction of GMOs can disrupt these delicate interactions. For example, genetically modified crops that exhibit increased competitiveness or resistance to pests may outcompete native species, leading to shifts in plant communities and potentially impacting other organisms that depend on them. Loss of biodiversity. The spread of modified genes to wild populations raises concerns about the potential loss of biodiversity. Genetic diversity is essential for the resilience and adaptability of species to changing environments. The introduction of genetically modified traits into wild populations may homogenize gene pools, reducing the genetic variability that is crucial for species survival and their ability to respond to environmental challenges. Addressing ecological risks. Addressing the ecological risks associated with genetic engineering and gene flow requires comprehensive strategies and precautions. Strict containment measures, risk assessments, and regulations are necessary to prevent the unintended release of genetically modified organisms into natural environments. Additionally, ongoing monitoring and ecological studies are crucial for evaluating the potential impacts of GMOs on wild populations and ecosystems. These assessments help identify and mitigate any unforeseen ecological disruptions, allowing for informed decision-making and responsible management of genetically modified organisms. Promoting sustainable agricultural practices, conservation efforts, and the preservation of natural habitats can contribute to mitigating ecological risks. By adopting ecosystem-based approaches and promoting biodiversity conservation, we can strive to maintain the integrity of natural ecosystems and safeguard biodiversity while ensuring the responsible use of genetic engineering technologies. Overall, by adopting precautionary measures, conducting thorough risk assessments, and prioritizing the preservation of natural ecosystems and biodiversity, it is possible to navigate the ecological risks associated with the introduction of genetically modified organisms and minimize any potential negative impacts on our environment. Health risks, considerations related to genetically modified organisms, GMOs. Genetically modified organisms, GMOs, have been a subject of scrutiny when it comes to potential health risks. While extensive testing and regulation are in place to ensure the safety of GMOs, there are considerations and ongoing debates regarding their potential impact on human health. 1. Allergenicity and unintended effects. One of the primary health concerns associated with GMOs is the possibility of allergenicity, meaning the potential to cause allergic reactions in susceptible individuals. Genetic modifications may introduce new proteins or alter existing ones, potentially leading to unintended allergenic substances in food or other products derived from GMOs. Rigorous testing and assessment are crucial to identify and mitigate such risks before GMOs are approved for commercial use. Furthermore, the complexity of biological systems introduces challenges in predicting all potential unintended effects that genetic modifications may have on human health. Despite rigorous testing, there is a need for ongoing monitoring and research to assess any long-term or subtle health impacts that may arise from the consumption of GMOs. 2. Antibiotic resistance. The use of antibiotic resistance marker genes in the development of genetically modified crops and organisms has raised concerns about the potential transfer of antibiotic resistance traits to bacteria in the gut microbiome. This transfer could contribute to the emergence of antibiotic-resistant strains of bacteria, reducing the effectiveness of antibiotics in medical treatments. As a result, minimizing the use of antibiotic resistance markers and developing alternative selection methods in genetic engineering are crucial considerations to mitigate this risk. 3. Unintended effects on nutrition and composition. Genetic modifications may impact the nutritional composition of genetically modified crops or food products derived from GMOs. Changes in nutrient levels, vitamin content, or the presence of antinutrients can have implications for human nutrition. It is important to conduct thorough nutritional assessments to ensure that the nutritional value and safety of GMOs are adequately evaluated and monitored. 4. Labeling and consumer awareness. Another consideration regarding health risks associated with GMOs is the availability of accurate labeling and transparent information for consumers. Individuals may have personal preferences or dietary restrictions that require them to be aware of the presence of GMOs in food products. Access to clear and reliable information empowers consumers to make informed choices based on their individual health concerns and values. 5. Contamination and coexistence. Ensuring coexistence and preventing the unintended presence of GMOs in non-genetically modified crops is crucial to address health risks and meet the demands of diverse consumer preferences. Strict regulations and coexistence practices are necessary to prevent cross-contamination during cultivation, storage, and distribution, ensuring that non-GMO products remain free of unintended genetically modified traits. Promoting safety and public health. Addressing health risks associated with GMOs requires a multifaceted approach involving thorough risk assessments, regulatory oversight, ongoing monitoring, and transparent communication. It is essential to ensure that the potential health impacts of GMOs are carefully evaluated, and that appropriate measures are in place to protect public health, support consumer choice, and promote the safety and well-being of individuals. Continued research, advancements in testing methodologies, and responsible regulation can contribute to a comprehensive understanding of the potential health risks associated with GMOs, enabling informed decision-making and the responsible use of genetic engineering technologies in relation to human health. Health risks, evaluating concerns associated with genetically modified organisms, GMOs. Evaluating potential health risks is a crucial aspect of genetically modified organisms, GMOs. Despite extensive testing conducted before their release, concerns persist regarding the long-term effects of consuming genetically modified foods and the potential allergenicity or toxicity of specific modifications. Addressing these concerns requires rigorous safety assessments, transparent regulations, and ongoing research. Long-term effects and safety of genetically modified foods. Questions have been raised regarding the long-term effects of consuming genetically modified foods on human health. Evaluating the impact of GMO consumption over extended periods is complex due to various factors, including individual variations, diverse genetic backgrounds, and dietary habits. Conducting comprehensive studies that consider these factors is necessary to assess any potential long-term health effects associated with GMO consumption. Allergenicity and toxicity. One concern involves the potential allergenicity or toxicity of specific genetic modifications in GMOs. Genetic engineering techniques may introduce new proteins or alter existing ones, which can lead to the production of substances that could trigger allergic reactions or have unintended toxic effects. Rigorous testing protocols are employed to identify and mitigate these risks before GMOs are approved for commercial use. Assessing the potential allergenicity and toxicity of GMOs requires detailed investigations, including laboratory testing and animal studies. Rigorous safety assessments. Safety assessments are a critical aspect of ensuring the responsible development and deployment of GMOs. Extensive testing protocols are in place to evaluate the potential risks associated with genetic modifications. These assessments include analyzing the molecular characteristics of the introduced genes, investigating potential allergenicity and toxicity, assessing the nutritional composition of GMOs, and evaluating any unintended effects on the environment or non-target organisms. Regulations to address health concerns. To address health concerns associated with GMOs, robust regulatory frameworks are necessary. These regulations vary across countries and regions but generally require comprehensive safety assessments, labeling regulations, and transparency in the release and marketing of GMOs. Strict adherence to these regulations ensures that potential health risks are thoroughly evaluated and that consumers have access to accurate information about GMOs to make informed choices. Ongoing research and monitoring. Continued research and monitoring are essential to evaluate the safety and health impacts of GMOs. Advancements in scientific methodologies and ongoing surveillance enable the detection of potential risks that may arise from genetic modifications. Monitoring the consumption patterns and health outcomes of populations can provide insights into the long-term effects of GMOs and inform future safety assessments and regulatory decisions. Public dialogue and engagement. Engaging in public dialogue and fostering transparency is crucial for addressing health concerns related to GMOs. By involving stakeholders, including consumers, scientists, policymakers, and advocacy groups, concerns can be addressed, and a better understanding of risks and benefits can be achieved. Open discussions and access to accurate information help build trust and promote informed decision-making regarding the use of GMOs. In conclusion, addressing health risks associated with GMOs requires a comprehensive approach that includes rigorous safety assessments, transparent regulations, ongoing research, and public engagement. By continually evaluating potential long-term effects, assessing allergenicity and toxicity, and implementing robust regulatory frameworks, we can ensure the responsible development and use of GMOs, prioritizing the safety and well-being of individuals and supporting informed decision-making. Socioeconomic considerations. Assessing the impact of genetic engineering. The application of genetic engineering carries significant socioeconomic implications that extend beyond the scientific and technological aspects. Understanding and evaluating these implications is crucial to comprehensively assess the potential effects on different sectors, stakeholders, and society as a whole. Implications on agricultural systems. Genetic engineering has the potential to influence agricultural systems, with both positive and negative socioeconomic consequences. One major concern revolves around the concentration of power and control in the agricultural sector. The widespread adoption of genetically modified crops may result in increased corporate dominance, limiting the autonomy and choices of farmers, particularly smaller-scale producers. Intellectual property rights and patenting issues related to genetically modified seeds can further exacerbate this concentration of power. Access and equity. Socioeconomic implications also arise regarding access to genetically modified technologies. While genetically modified crops can offer advantages in terms of productivity and resilience, their affordability and accessibility may pose challenges for resource-constrained farmers, especially those in developing regions. Ensuring equitable access to genetically modified technologies, particularly for smallholder farmers, is crucial to prevent further inequalities in the agricultural sector. Impacts on rural communities. The socio-economic impacts of genetic engineering extend to rural communities. Changes in agricultural practices and the adoption of genetically modified crops can affect livelihoods and traditional farming practices. It is important to assess these impacts and ensure that rural communities are engaged, empowered, and supported throughout the process of adopting genetically modified technologies. Promoting participatory approaches, knowledge sharing, and capacity building can aid in maximizing the potential benefits for rural communities. Consumer perception and market demand. Public perception and consumer attitudes towards genetically modified products can influence market demand and have socio-economic implications. Concerns over food safety, environmental impacts, and ethical considerations may affect consumer acceptance and willingness to purchase genetically modified products. Transparent labeling and clear communication of the benefits and safety measures associated with genetically modified organisms are crucial to enable informed consumer choices and maintain market confidence. Economic growth and trade. The adoption of genetic engineering can impact national economies and international trade. Countries with advanced genetic engineering capabilities may experience economic growth through innovation, increased agricultural productivity, and export opportunities. However, trade barriers, regulations, and divergent policies regarding genetically modified organisms can create challenges in international trade and market access. Harmonization of regulatory frameworks and international agreements can help mitigate these challenges and facilitate trade. Ethics and societal values. Genetic engineering raises ethical and societal value-based considerations that can have socio-economic implications. Diverse perspectives exist regarding the manipulation of genetic material, patenting of genetically modified seeds, and the potential impacts on ecosystems and biodiversity. Engaging in ethical debates, public consultations, and involving stakeholders in decision-making processes can help ensure that the socio-economic implications of genetic engineering align with societal values and ethics. In conclusion, assessing the socio-economic implications of genetic engineering is essential to understand the broader effects beyond the scientific and technological advancements. Evaluating impacts on agricultural systems, access and equity, rural communities, consumer perception, market dynamics, economic growth, trade, and societal values is crucial for informed decision-making and responsible implementation. By addressing these implications and promoting equitable access, participation, and sustainable practices, genetic engineering can contribute to socio-economic development while safeguarding societal well-being and values. Socio-economic concerns, implications of widespread adoption of genetically modified crops. The widespread adoption of genetically modified crops has given rise to significant socio-economic concerns, particularly regarding the control over agricultural practices, seed supply, and the potential impact on small-scale farmers and traditional agricultural systems. Corporate control and agricultural practices. A primary concern is the concentration of power and control within the agricultural sector due to the dominance of large corporations over genetically modified seeds. These corporations often hold intellectual property rights and patents on genetically modified crops, granting them control over the production, distribution, and pricing of these seeds. This concentration of power can limit the autonomy and choices of farmers, especially smaller-scale producers who may face challenges in accessing alternative seed varieties or competing in the market. Seed supply and diversity. The control of genetically modified seeds by large corporations can also impact seed supply and diversity. Traditional seed-saving practices may be discouraged or replaced by a dependence on commercially available genetically modified seeds. This shift can lead to a loss of traditional seed varieties, undermining agricultural biodiversity and potentially reducing resilience in the face of changing environmental conditions. Preserving and promoting access to diverse seed resources becomes crucial to maintaining sustainable agricultural systems. Impact on small-scale farmers. The adoption of genetically modified crops may have differing impacts on small-scale farmers, who often have limited resources and face unique challenges. While genetically modified crops can offer advantages such as increased productivity or resistance to pests and diseases, the economic feasibility and affordability of genetically modified technologies can be a barrier for small-scale farmers. In some cases, the adoption of genetically modified crops may require significant investments in seeds, equipment, and associated inputs, posing challenges for farmers with limited financial resources. Traditional agricultural systems and cultural heritage. The widespread adoption of genetically modified crops can potentially disrupt traditional farming practices and impact traditional agricultural systems. These systems often embody local knowledge, sustainable practices, and cultural heritage that are deeply intertwined with the identity and livelihoods of rural communities. The introduction of genetically modified crops may lead to changes in cropping patterns, reliance on external inputs, or alterations in farming techniques, potentially challenging the sustainability and resilience of traditional agricultural systems. Empowering small-scale farmers and preserving diversity. Addressing the socio-economic concerns associated with the widespread adoption of genetically modified crops requires initiatives to empower small-scale farmers, promote equitable access to resources, and protect agricultural diversity. Supporting sustainable agricultural practices, providing technical assistance, and facilitating access to diverse seed resources can help safeguard traditional farming practices, preserve cultural heritage, and foster resilience in agricultural systems. Promoting transparent and inclusive decision-making. Transparent and inclusive decision-making processes are essential to address the socio-economic implications of genetically modified crops. Engaging farmers, local communities, civil society organizations, and policymakers in discussions and policy development allows for diverse perspectives to be considered. Such inclusive processes can help shape regulations, promote equitable distribution of benefits, and ensure that the adoption of genetically modified crops aligns with the needs and values of different stakeholders. In summary, the widespread adoption of genetically modified crops raises socio-economic concerns related to corporate control over agricultural practices and seed supply, potential impacts on small-scale farmers, and the preservation of traditional agricultural systems. Addressing these concerns requires promoting equitable access, protecting agricultural diversity, empowering small-scale farmers, and fostering inclusive decision-making processes. By balancing innovation with social and economic considerations, genetic engineering can contribute to sustainable agriculture and inclusive rural development. Importance of ongoing debate and responsible application of genetic engineering. Ongoing debate and research surround the advantages and disadvantages of genetic engineering, highlighting the need for careful consideration and responsible application of this technology. By addressing potential risks, ethical concerns, and maximizing benefits, genetic engineering can be utilized in a manner that promotes societal well-being and sustainable development. Advancing knowledge and understanding. The ongoing debate and research surrounding genetic engineering contribute to advancing our knowledge and understanding of its potential impacts. Through scientific investigations, societal dialogue, and interdisciplinary collaborations, we continually strive to gain deeper insights into the advantages, disadvantages, and complexities associated with genetic engineering. This ongoing process of learning and discovery helps inform responsible decision-making and ensures that scientific advancements are guided by ethical and societal considerations. Balancing benefits and risks. maximizing the benefits and minimizing the potential risks of genetic engineering is a key objective. While genetic engineering offers promising possibilities, it is essential to carefully evaluate and mitigate potential risks. Responsible and regulated application of genetic engineering techniques involves rigorous risk assessments, transparency, and robust regulatory frameworks. By striking a balance between innovation and precaution, we can harness the advantages of genetic engineering while minimizing any adverse effects. Ethical considerations and public perception. Ethics play a crucial role in the ongoing debate surrounding genetic engineering. As we explore the potential benefits and disadvantages, it is essential to address ethical concerns associated with manipulating living organisms and the potential consequences for ecosystems, biodiversity, and human well-being. Engaging in ethical deliberations, incorporating diverse perspectives, and ensuring transparency in decision-making processes are essential for promoting public trust and maintaining the social license for genetic engineering technologies. Regulation and oversight. The responsible application of genetic engineering requires robust regulation and oversight. Governments, regulatory agencies, and international bodies play a crucial role in establishing frameworks that ensure the safe and ethical use of genetic engineering techniques. These frameworks include thorough safety assessments, labeling requirements, monitoring programs, and public engagement mechanisms. Continuous evaluation and adaptation of regulatory measures are necessary to keep pace with scientific advancements and evolving societal concerns. Sustainable and inclusive approach. A sustainable and inclusive approach to genetic engineering is vital. This involves considering the long-term impacts on ecosystems, biodiversity, and social well-being. Assessing the potential benefits and disadvantages through a sustainability lens allows us to evaluate the environmental, economic, and social dimensions of genetic engineering. Inclusive approaches involve engaging diverse stakeholders, including scientists, policymakers, farmers, consumer groups, and civil society organizations, to ensure that decisions reflect a wide range of perspectives and considerations. Education and public awareness. Promoting education and public awareness is crucial for informed decision-making and fostering constructive dialogue. By providing accessible information about genetic engineering, its potential benefits, and the associated risks, we can empower individuals to form well-rounded opinions. Public engagement initiatives, educational programs, and transparent communication channels help bridge the gap between scientific advancements and public understanding, fostering trust and allowing for informed public input. In conclusion, the ongoing debate and research surrounding genetic engineering reflect the commitment to responsible and ethical application of this technology. By addressing potential risks, ethical concerns, and societal considerations, we can harness the benefits of genetic engineering while ensuring the well-being of individuals, the environment, and future generations. Ongoing dialogue, robust regulation, and a sustainable approach guide us towards the responsible and beneficial utilization of genetic engineering for the betterment of society.